Good afternoon, everybody, and welcome to this departmental seminar of CBS department. And it's a pleasure to have with us today Dr. Supratim Banerjee from IISER Kolkata. And it's a particularly pleasure for me because I know Supratim for a while. We were batchmates during our integrated PhD program at IISc. So Supratim did both his master's and PhD from IISc Bangalore, in the Department of Organic Chemistry. So he completed his PhD in 2011, and then he did two postdoctoral stints in Germany, one at University of Regensburg and University of Duisburg-Essen in Germany. And then he joined Department of Chemical Sciences in IISER Kolkata in 2016, and currently he is associate professor there. And his research interests include luminescent supramolecular polymers, photoresponsive materials, small molecule and multivalent luminescent sensors for bioanalytes, host-gas chemistry, artificial light harvesting systems, and room temperature organic phosphorant materials. So today he's going to talk to us about aqueous self-assembly of chromophore appended amplifiers, bioanalyte sensing, photoreactivity modulation, and energy transfer studies. Super.
It is okay, huh? I guess not fit. So can you give the PowerPoint slider?
Yes, sir. Yes.
Okay, let me just first thanks Sumon and also for hosting me and for giving a kind of chance to come here. As he mentioned that we know each other for quite some time now, but recently we started kind of a little bit of collaborative work, which is very preliminary stage. And I thought this is a good time, and he mentioned that I can visit here and give a kind of presentation on some of the recent things that we are doing in our group. So as you can see that, we work on, kind of molecules. If you think that generally we are going through many things about the title that we work on functional organic materials. But these materials we try to design in aqueous media so that you can use them for different purposes. And as you see the term self-assembly, and I think all of you know that the self-assembly has emerged as a kind of nice protocol for making different kind of molecules with different functions. But when you go for aqueous self-assembly, moving for something in the organic media, you need to have a kind of specific kind of different design than what you employ in organic media. One of the problems of making organic functional materials in aqueous media is that these organic molecules are typically hydrophobic. And when you bring to the water, it's sometimes difficult to control their aggregation. What you do, you try to modulate the structure such a way that you bring a kind of solubility to them, but you also have other groups which will actually allow them to form self-assembly. And what you see here, chromophore appended amplifiers, so you work on the molecules which have both polar and non-polar parts. I mean, amplifier is a very common term which has been used for a long time for molecules which can aggregate in water by using the non-polar groups. And we work on chromophore appended, and this chromophore actually we work on different kind of chromophores. I will show them very soon. Today's talk I will focus on these three topics. How we use these aggregated structures for bioanalyte sensing. We can also play with the photoreactivity in these structures, and then energy transfer studies to external dyes when they are incorporated in those assemblies. So if you look at, as I have already mentioned, we are interested in organic aqua materials, and we do something called host-gas chemistry, where we bring macrocyclic hosts to modulate the chromophoric properties. We work on these things, and we have worked on a lot recently, self-assembled multivalency bioanalyte recognition, where we use these nanostructures. We have the kind of charges on their surfaces to bind to bioanalytes. We work on supramolecular assemblies and polymers. The other subgroup of this thing is photoresponsive materials, where you can irradiate light on the self-assembled structures and modulate their properties, mainly the luminescence. And the other two areas also we work on artificial light harvesting, and something we have started more recently, room temperature phosphorescence from organic materials. So as you see from this design that we typically start from organic building blocks, and then we make kind of assembly in aqueous media, use them for different purposes. So synthesis is very important. But today's talk, I will not show any synthetic slide. You have to assume that we have made these molecules in the lab, and then I will just talk about the application of this molecule. So we work on both kinds of chromophores, both in the sense means this is my kind of classification. These are more traditional chromophores, anthracene and pyrimine, and these which are planar chromophores. These are what you have twisted chromophores. You see that they have a conjugated structures, but as I will talk very soon about them, that they have a kind of twisted nature. We actually connect them to the amphiphilic, like make amphiphilic molecules out of them, and then we look for their self-assembly properties. So before going to the three topics that I talked about, just go through some of the things that we have been doing recently. So as I mentioned, that we use host-based chemistry. These are something like which you use regularly to decrease the macrocyclic host molecules. The oligomeric molecule, you can see, you can vary the number N here, and these molecules are very interesting because they are soluble in water, but they have hydrophobic pocket. So if you put the chromophoric molecules inside the pocket, there will be a modulation in the luminescence property. So we have used that for different projects. The other thing which I have already mentioned, that we target this kind of bioanalyzed separate ATP DNA. If you look at them, you see that all of them have these negative charges, because if you look at our molecules, which I have shown few slides earlier, all of them have actually positive charge in their back. So when you generate the self-assembled structures, they have what we call a lot of charges on the surface, so they can attract these biomolecules. And especially for some of the molecules like this molecule, which has a double bond, cyanosilbine, we can also have a photoreactivity like a carbon-carbon double bond photoreactivity, and we are exploring different photoreactions again to modulate the properties. Also, when you have these, kind of co-assembly formation between heparin and the self-assembled structure, the co-assemblies we have used for kind of fluorescent resonance energy transfer studies. And the other thing that I have already mentioned, that we are also working on this room temperature phosphorescence starting from organic materials, and this is also something which is a very rapidly growing field in the last few years. So when you want to have phosphorescence and room temperature, it is typically obtained through organometallic compounds or inorganic complexes, because they have a very strong intersystem cross. In organic materials, purely organic materials, it is a challenge. Today, I will not talk about that, but we are working, a couple of students are working on that aspect also. So today in my talk, in several projects, you will see this molecule coming back. So let me give a very small introduction there. What are the special features of this molecule? So this is called cyanostilbene. So all of us read stilbene, and when you replace one of the hydrogens here in the carbon-carbon double bond by a cyano group, this is called cyanostilbene. When you look at the structure, you feel that that is just a very small modification. But what happens once you introduce cyano, which is not visible from this image that I am showing, but hydrogen and cyano, they actually kind of spherically repel each other. They don't want to occupy the same space. So what happens? There is a kind of rotation around this bond. So if you build a cyanostilbene, which was reported 20 years back, where you can see that you have this cyano group here as well as the biphenyl. And if you look at a simple biphenyl system, what you have read in our basic chemistry, the biphenyls are generally having a kind of twist structure. So if you try to kind of draw a resonance structure, you will see that you can actually delocalize the electrons from this end to that end. But in reality, what happens? You have all these twists along the backbone, so the molecules are not completely balanced. So what is the effect? Its effect is that when you take in a solvent where the molecule is completely dissolved, what we call monomeric state or molecularly dissolved state, and you try to excite these molecules by some light which actually it can absorb, the excited state actually loses the energy. Because you have all these, twisted structure, they have this torsional motion. So excited state energy, rather than coming as an emission, you lose it or you spend the energy in this torsional motion. One way to stop this, if you actually induce aggregation, like in this molecule they added water and they produce this aggregate. In the aggregate, as you can understand, many molecules are packed together. Though the packing is not good as really the planar chromophore, but nonetheless, it actually stops many of these non-radiative deactivation. So when you go from a solution molecularly dissolved state to aggregated state, you actually gain in emission. So if you see the recent literature, many people call this what we call aggregation induced emission, but this term has its own, kind of, some people don't like the term, but at least for our purpose, we are happy that the molecules we designed, they actually showed high emission in the aggregated state. I must mention that what is the importance, because if you take planar molecules, planar chromophores, if you bring them close proximity to each other, many cases there is something called self-quenching or concentration quenching. So many of these chromophores are very highly, luminescent in solution in monomeric state. Whereas you bring kind of many of them close together, either in high concentration or in the solid or aggregated state, then they lose the fluorescence. And many of these applications, when you want to make a film or solid coating of these molecules, you have to bring many molecules together. This is where this kind of molecules which have twisted structures are interesting, because they can actually gain in their luminescence. So we wanted to, as I told you, we wanted to make something which will be, kind of compatible in the water media. So what we did, we took this kind of cyanosilbine, but at the end we added this kind of pyridinium group. So what it does now, it introduces two charges at the two ends of the molecule, and there is this cyanosilbine chromophore, and we can actually add different kind of R groups, simple alkyl substitution. So that what happens, because of the two positive charges, it will go into water, but then if you keep on increasing the alkyl group length, at some point it will also start aggregation, because the hydrophobic groups will actually tend to make them aggregated, and in the aggregated state, cyanosilbines will come close together. They will have length or non-radiative decay, and finally you will have more luminescence. So just to illustrate that, once you design this kind of molecule, they have actually kind of versatile properties. Let me highlight. So I have already mentioned about the tuning of the hydrophobicity by introducing this aliphatic chain, and you can do work-based chemistry to modify or modulate the fluorescent properties of these molecules. You have this pyridinium, so these positively charged groups give them the chance to use them for anionic bioanalysis sensing. And finally, if you look at the carbon-carbon double bond, you can do a photochemistry, several kinds of reaction, which will again modulate the chromophoric structure. So once you generate this kind of molecules, this can be used for various purposes. And in today's talk, I will highlight some of the things that we have been able to achieve in the last few years. So what we do in the lab, so you want to probe the cell assembly, you need a kind of technique that will tell you that the molecules are undergoing cell passage. So one of the things that we use in optical spectroscopy, absorption and fluorescence spectroscopy. So here, what you see that two cyanosilbines are taken. This is having a small alkyl chain C2. This is C12. So this molecule was nicely soluble in both water and DMS. This molecule, on the other hand, soluble in DMS nicely, but not in water. So when you look at their absorption spectra, and we started from DMSO, they started adding more amount of water. What you see for the small molecule C2, which does not undergo aggregation under this condition, so there is a just small shift in the absorption spectrum. But when you go for the C12, especially at 90 and 99%, you see that there is a decrease in the absorption, as well as there is a shoulder band coming up, and these things told us that it is undergoing aggregation. When you looked at the emission spectra, there is a very drastic change in the emission spectra. So in DMSO, it shows an emission around 460, 450 nanometer. And as soon as you go to higher water content where it started aggregating, you see a new peak comes out. This is a broad peak, it's close to 540 nanometer, and it has a much longer lifetime than what you can observe for a cyanosyl beam, which is molecularly dissolved state. So the experiments told that it is excimeric in nature. And those who work on this kind of molecules, so excimer basically means you have an excited state molecule interacting with a ground state molecule. Energy level is slightly lower, so you see an absorption, I mean the emission at higher wavelength. Of course, we did other studies to show you can see in SEM very nice one-dimensional nanofibers. You can do dynamic light scattering studies which show the formation of nanostructures here. So based on that, we thought that we'll try to use this for multivalent binding of analytes. So what is multivalency? This is sometimes, yeah, it's okay. Is it continuing correctly? Sometimes I feel that it is going.
Put it on the other color. On the other side.
Yeah. Can you just put it? Hello. Oh, thank you. So multivalency is a concept which is used in biology a lot, and all of us last 2.5 years we are looking at this deadly virus. So if you look at the virus, they are having a lot of protein spikes on the surface, which allows this virus to go and bind to the host cell. So that is the perfect example of multivalency. That means rather than 1:1 interaction, you use multiple binding sites from both the partners, and which is enthalpically and entropically very strong. So multivalency is a technique which is used in biology a lot. We thought that when you generate these nanostructures, for example, for these long alkyl chain cyanosyl beam, they generate nanofibers. I have shown some other cases they generate other structures. You will have a kind of multivalent array of charges on the surface, and because the backbone is positively charged, you have a positively charged surface. So if you bring anionic analyze, it should be bind. It should bind to the surface. So we looked at heparin. Heparin is something which is used regularly as anticoagulant. You see the structure. This is a sugar-based molecule. This is a typical monomeric unit with a 2 sugar units. And what you see that there are 4 groups which are all deprotonated at physiological pH. So heparin is actually the most charged dense molecule, biomolecule known. It's much more charged dense than DNA. So what we thought, because it has a very high charge density, and then if we bring this positively charged nanostructure, they will have a strong multivalent interaction. And why we looked at heparin? Because it is used as an anticoagulant. That means when you need the blood not to clot, you can add heparin. And there are many complications when you use heparin. Source, depending on the source of heparin, its activity varies. Its effect also on patients varies from one person to another. So sometimes the exact quantification of heparin is very important, and that's why in recent years people have tried to make kind of fluorescent sensors which can detect heparin, in competitive medium. So what we thought that we'll use this cationic cyanosyl beams for this purpose. So as I have already mentioned, that depending on the chain length, they can either be in the aggregated state in aqueous media, for example 8, 10, and 12. When you go to lower alkyl chain, 2, 4, or 6, they don't aggregate because the aggregation is also induced by this hydrophobic chain. But what we found interestingly that whether you started with the aggregated sample or the smaller groups, both of them actually show heparin detection. In this case, it is more like a multivalent binding where these large nanostructures are binding to the heparin. The other case, what happens, these monomeric compounds, 2, 4, 6, they can form small clusters on heparin surface, but both of them actually have this strong excimeric division. So I will not talk about this part. Let me talk a few things on this multivalent heparin binding. So how do you know that the multivalent binding is heparin? There are a few ways to look at it. One thing is that, for example, you see that this is a size before adding heparin because it is already self-assembled structure. But when you add heparin, now there is extensive aggregation, so you see an increase in the size. And other thing that actually gives this kind of indication is zeta potential. So all of you know that the zeta potential is something which is the surface excess surface charge. So when you have this positively charged self-assembled structures, you have a positive beta potential. If you keep on adding heparin, you go high above concentration of heparin, you can actually have negative beta potential, and you can understand now that heparin is actually binding to this active. But the way we detected heparin is through fluorescence chain. As you see, this is before adding the heparin, and if you start adding heparin, you see a huge increment in the fluorescence. The wavelength does not shift, so that tells you that the chromophore packing is probably not too different. But what is happening once this electrostatically driven co-assembly formation happens, you have actually probably much more tighter packing. And what you see here also that the quantum yield after heparin addition and also the lifetime, both of them actually increase.
I have a question.
Yeah.
Binding is mostly electrostatic since you are seeing the charge inversion. So, is it possible that you add some negatively charged any molecular atom and you will see the charge inversion, like the any negatively charged phosphates or something?
Phosphates or carboxylates, typically what happens, because the heparin has a very strong charge, high charge density. So adding a negatively charged thing is difficult because then you have to go beyond heparin in terms of the charge density. Because what we have figured out, we have also done this study with polyacrylic acid, which has only one negative charge. That also binds, but the binding is not as strong. I mean, we have not estimated the exact value. But what we have done, actually, that I will not show today, that there is something called protamine sulfate, a kind of arginine-rich peptide, which is used for heparin anti. So that means that we have done. So we have seen that this binding actually is stronger than protamine has. That completed experiment we have done, but they are not showing. So it is possible, but at least with the analytic thing, it's a bit difficult because you need that kind of charge and this.
So might be some specific interactions also present.
Yeah. So as you mentioned, what you figured out that there is this hydrophobic residues also playing a role because it is ultimately a co-assembly. Electrostatic force, of course, is the main driving force, but these hydrophobic chains also play a role. That we have seen when you look at the different derivatives, because all the derivatives do not show the exact equal sensitivity in binding. Some of them are better. So typically the higher alkyl chain here shows better sensitivity. So that is okay. So these studies were all done in aqueous buffer at 7 point, which is good, physiological quantity. But if you really want to use heparin and for heparin sensor, you have to do in plasma and serum. Plasma and serum, the difficulty is that they contain high concentration of electrolytes and also some protein residues. So what happens when you put this and you think about, as he already mentioned, that is the electrostatic binding between negatively charged heparin and positively charged surface. If you have a high salt concentration, this will screen this electrostatic. So typically, if you see literature, other than few examples, most of the people do this in 5-10% serum or plasma. So you take the pure serum or plasma from the patient or the person for whom you need the heparin, you want to estimate the heparin in blood, and then you dilute into buffer. Otherwise, you do not see the sensitivity. But what we could figure out that in our case, because whatever this multivalent nanostructure, we could go to 75% human plasma and 50% human serum. So you could go very high percentage of serum and plasma, where we could still detect heparin in medically recommended concentrations. So if you see this work, this was mainly done for the detection. But then when you looked at this co-assemblies, because we had many other cyanosilmic derivatives, we thought, okay, once you make these co-assemblies between heparin and these positively charged nanostructures, you see that there is a huge increment in the process. We thought that can we actually transfer this excitation energy to other external dyes through resonance energy transfer. So as you know, energy transfer is very important in natural photosynthesis, where sunlight is initially captured by all these antenna chromophores, and then they reach the reaction center through many energy transfer states, which are of course very highly efficient. So people have been working. There are many strategies and all. I will not talk about that. Very nice examples of artificial light harvesting. And one of the main mechanisms that people have observed is something called Foster mechanism, where you excite a donor molecule, and then there's through this excited state dipole can actually excite another acceptor molecule, but there is no photon getting transferred between them. So this is called dipole-dipole mediated interaction, and is called Foster mechanism. So what we thought that we can use some of these things for that, because in recent years we have observed that recent years, in maybe last 2 decades or so, that among other methods, one of the methods people have used is a biotemplate based light harvesting. You take DNA or DNA based nanostructure, maybe virus or proteins, and where you arrange different chromophores and you look at energy transfer. Why energy transfer is important? Because it is not only useful to know the basic things, what is happening in natural photosynthesis, but it is also useful for design of solar cell, for building photocatalysts and many other systems. So energy transfer is an actually interesting phenomena in terms of fundamental understanding, increasing the fundamental understanding, and also for application purpose. So in our case, we took heparin and this cationic and still means the higher analogs, which, as I have told, that they form larger co-assemblies with heparin, and as I have already mentioned, there was an increment in the fluorescence. The other thing that we have observed at this moment, I will not talk much about that, that the initial self-assembled structure only from these molecules actually from nanofiber. That means you have a solid nanofibers, we get converted to kind of nanotubes when you add heparin. So there is a change in morphology also. But we are more interested to see these nanotubes when you actually add external dyes, whether they can go close to this nanotube or get integrated in the nanotube structure. Because you remember this is often from self-assembly or co-assembly. So there is a chance that the dyes can go and get close to these donor molecules. The donor molecules, of course, will be the cyanosilvanes. And what we did, I mean, this is not all of them probably not visible, but we have investigated different dyes. But today's talk, I will concentrate on Nile red. So Nile red is a very well-known dye which people use. If you take Nile red in aqueous solution, it is typically having very low emission. It gets quenched, but once you put it in the hydrophobic pocket, our environment is emission increases. And why we chose these dyes? Because we figured out that all of them have a nice spectral overlap, what is called between the emission spectra of the donor that is coming from the cyanosilvane in the co-assemblies and their absorption spectra. This is one of the basic requirements for performing FRED experiments. So I'm not showing all these values, but we have calculated and we have seen that all of them actually have decent overlap. But today I will talk about the Nile red because it showed the best results out of all these dyes. So when you look at energy transfer, you look at mainly two parameters. One is energy transfer efficiency, which you can measure many ways. One is that how much donor emission is getting quenched when you keep on adding the acceptor. And other thing that how much gain you have in the acceptor emission. So this is, for example, the acceptor emission when there is no energy transfer. But when you have an energy transfer, now you see an amplified emission from the acceptor. So the ratio of this actually will give you the antenna effect. So both are important because if you have a nice energy transfer efficiency, that should also lead to a reasonably high antenna effect. And these are the experiments we did, and you see that we have these luminescent nanotubes made from this 12C and heparin co-assembly. Then we add Nile red, they go sit somewhere in the NAS structure. We have done experiment to show that they are getting integrated in the structure. And what you see that initially the emission before adding Nile red was in the greenish yellow range. And this thing is not very visible, just follow my writing that if you keep on increasing the Nile red amount, you see that the acceptor emission keeps on increasing because the efficiency is also dependent on how much Nile red you can do. But you can see here that we could achieve a very good energy transfer even at 1 mole% doping of the acceptor. So this is quite good, especially in the aqueous media. And I will not go too much into the number, but we could measure these numbers, which are quite good for this aqueous-based system with Nile red. So we also did a control. Control means this molecule, even without heparin, also forms self-assembled structure, just to see whether these are also efficient. So they also showed energy transfer efficiency, but you see this value of 52% is lower than what we could get with the co-assembly. So the co-assembled structures actually were better energy donor compared to the only self-assembled donors. So I'll talk about another one, which is again on FRED, but this is based on a different molecule. So far I have talked about cyanosyl bit, but as I have mentioned in the first few slides, that we also work on amplifiers which are derived from these more classic chromophores like pyrene. And what we have seen that these chromophoric molecules, these amphiphilic chromophores also, when they form self-assembly, they show excimeric emission in aqueous medium. So again, the main principle is same, that you bring external dyes and you try to see energy transfer from the excited donor to the acceptors. And in this case, although what we saw that the Fred process was multi-stimuli responsive, and I will talk about that now. So these are the molecules that we employ. Again, you see the design principle is more or less same. We vary these alkyl chain length, and when you have 8, 10, and 12, you see they started aggregating, they started showing this strong excimeric emission coming from pyrene around 490 nanometer, and you could see the aggregation through DLS and electron microscopy also. Then, when you add ATP, the structure is shown here. You see that ATP is not like heparin. It is a small molecule, but it is also having a very high charge density. So if you look at the pK of ATP, you will see that almost all the charges are actually deprotonated more or less at physiological pH. So there are, so when you think about ATP, it has 4 minus charges, and they are also in close proximity. So we expected when you add this ATP to these, positively charged nanostructures, which are made from the self-assembly of these molecules, they will also bind to them. And what we found again, that the binding was followed by a very strong increment in the excimeric emission, and which varied with the ATP concentration. One of the derivatives C10 showed the base value, so we carried on with C10 for the further studies. So what we did again here is that looking at the energy transfer values in the self-assembled structures without ATP and with ATP, and we found that when you have ATP, the energy transfer was much better, and this you can follow from this simple plot. As you see that this is a donor acceptor ratio of 200:1. So if you don't add ATP, zero concentration of ATP, that means you are only looking at the self-assembly. And if you bring 9 late, they also do energy transfer, but which is within 25 to 30%. So it's not very efficient. As soon as you start adding ATP, the co-assemblies form, you can actually reach values which are close to 90-95%. So there may be many factors, but one of the things that we have observed that when you calculate these overlap integrals, we found that the overlap integrals are typically higher for the co-assemblies compared to the self-assembly. And that is one of the reasons why we see higher assembly. But as I told, what we are doing, we are using external dyes to the structures, and how these dyes are introduced in the structure, that can also vary from co-assembly to self-assembly. The exact reason is difficult to tell, but we found that the co-assemblies were better, energy donors in the Fred studies. And the interesting part of these studies was that not only in the solution where you can vary the color. So you start from a cyan blue kind of emission, you add 9 lead which emits in the red region, so you can vary the color because you are mixing 2 colors now. You can go from blue to white to kind of pinkish red. And you can also actually make them as a kind of film. This is in some kind of polymer, polyvinyl alcohol. So this is having aqueous phase polyvinyl alcohol, but if you dry, you can still see that you can retain more or less the same colors. So this is not only useful that energy transfer in the solution phase, but also you can translate the film state. And what we figured out that this Fred process here in these ATP-based co-assemblies were also modulated by different stimuli. For example, if you look at this co-assembly, this is a co-assembly which is obtained from C10 and ATP. If you keep on increasing temperature, this is the excimeric emission from pyrene. As you can understand, when you go to higher temperature, there are 2 effects. Typically, a fluorescent molecule, if you take to higher temperature, it loses fluorescence because there are more non-radiative pathways. But what we are doing also at higher temperature, these co-assemblies also will start disintegrating. So there is a kind of dual effect. So you see, in going from 20 degree, which is here, you have a decreased emission. And then when you cool the system again from 70 to 20, you regain the emission. It's a very, very reversible process, which is shown here. So what will be the effect on the Fred? You understand this is the part of the emission which is actually kind of transferring the energy to the acceptor. So by increasing the temperature, if you decrease, then it will also have a result on the freight, and that is what we saw, that when you go to the higher temperature, the freight also diminishes. And interestingly, if you plot this two emission, this is the emission coming from Nile rate, this is the emission which is coming from the monomeric region of the pyrene, and you see that that is almost following a linear plot. This kind of things actually what you can term as ratiometric thermometers, because they can sense temperature, and if you see carefully, we are also covering this physiological temperature range, and you can also denote that change in the CIE diagram where the point moves from here to there when you go to lower temperature. We also found that if you have metal ion, which also selectively quench the emission of the excimer, again, we can vary the freight. I will not go too much into that. And also the pH, because as you can understand, that our coassembly is built between ATP and the positively charged structure. If you keep on going to lower pH, what will happen? It will protonate the ATP at some point. ATP will lose the charge, so coassembly is broken because electrostatic force will go down. So again, you will see a change. For example, if you see that this is in the presence of Nile rate, if you keep on adding HCl at some point, you will see that the freight goes down, so you can see only the emission from the donor, and you can gain back again by adding sodium hydroxide. Interestingly, you could also perform that in the solid state. So this is where you see there is nothing visible. This is actually Nile rate was plotted here, and this we call is a kind of encrypted information, which I will explain now. If you add the coassembly now, you see immediately a color generation even after drying. So the photos here are all are shown after drying, but you understand we are adding the aqueous solution and then we keep for some time to dry. And if you add HCl now, you kill the freight process. So you see the emission only from the pyrene amplified. You can get back again by dilute derivative. So if you think about something which is invisible here, this is actually 3-layer encrypted, because you can see 3 different colors by using 3 different students. So we have not drawn any letters and all, but this is a kind of application in the solid field. So this is what we can achieve with the project, that not only in the solution, we could see multi-stimuli responsive freight also in the solid state. Now I will come to the last part of my talk. This is a little bit different in a sense, because I will talk about a reaction in this assembly. And you know, when you look at cyanostill, because of the presence of this double bond here in this structure, they can undergo a number of photoreactions. So all these photoreactions are known for still beams and also for some cyanostill beams. So what happens? You have a kind of trans linkage here we call Z, because this is in the nomenclature. Then it goes to the E form or the cis form, and then it can undergo a kind of electrocyclization. So you see that this is, if you look at carefully, this is 6-pi electrocyclization, and this molecule is not very stable under ambient condition, that gets irreversibly oxidized to this cyclized form, which is a kind of phenolic. If you go back to the basic organic chemistry, this is the other analog of ancestry. So what you are doing here, you are starting from this trans cyanostill bean, and you can actually go through cis cyanostill bean and finally to this phenolic molecule. So this will actually modulate the luminescent properties. You can also do this 2+2 photocycle addition, where the 2 double bonds, if they are in a correct orientation and in close proximity, they can undergo this reaction. So we wanted to investigate that by changing the aggregation, by changing the nature of the cyanostill bean groups, can we actually modulate this reaction? So for that reason, initially we chose these 3 molecules. These 2 basically represent the amphiphilic version. They form aggregate in water, as I have already shown. N2 is again a control molecule which is soluble in water. So first we did the photoreaction of this molecule C2 analog. You see when you irradiate with light 365 nanometer, so the light I have not written, but what you see that the 360 nanometer band goes down, and you see the emergence of a new band in the absorption spectra. That tells you that one species is getting converted to another one, and there was a change in the emission also. We also saw similar absorption spectral changes in DFSO, and then we did an NMR analysis. I will not go too much detail into that, but what you can see here is that at 0 minute you have only the transform, and then when you keep on irradiating, you can see the new peaks coming up. This is for the cis compound, and when you do the irradiation for long enough time, you see the cyclized product also forming. So when we compare for this C2 compound in DMSO and D2O, what you see is that if you see this, this is the unreacted starting material. So after 3 hours of photoirradiation, when you look at the result from the water-based system, that the reaction was much faster with water because more starting material was converted. And also, if you look at the ratio, here the cis product is more, cyclized product is less. Water, this is actually higher. So though this was not very clear, but it is known in literature that when you do photoreaction of stearates, typically the reaction rate is faster in more polar solvent, and there is a reason behind that. I am not telling at this moment, but it is well known. Then we carried on the photoreaction on these two compounds. Remember, now these two compounds are in aggregated state, but the absorption spectral changes are very similar to that, small derivative C2. But now there is a change in the emission because the initially the emission was around 540 nanometer, is a greenish yellow emission, and then when you start doing photoirradiation, you start generating a cyan blue component which keeps on growing. So at this point, you generate a white light emission. Here, everything is looking yellow, but just assume that. I'm sorry, but so that is actually a nice white emitting sample, and finally to a very strong emitting cyan blue. So what we figured out is that that when you are doing the reaction, it generates these two cis compound as well as the cyclized product. And when you do in the aqueous media with a small compound, which is a molecularly dissolved state, the ratio of CY and E was 1.40. But when you do the same reaction in this, what you call supramolecular polymer or assembled state, you see a much higher proportion of the cyclized product. And this is actually known for some of the twisted molecules in the solid state. Because if you think about the twisted molecule, and if I go back to the photoreaction, let me tell you. So this product, which is finally formed, this goes through this intermediate. So this intermediate formed from the cis structure, which is formed first after the irradiation of the trans compounds. And when you want to have this electrocyclization here, you understand that these 3 pi bonds, they should have a little bit. I mean, the carbons which are holding the pi bond should have some kind of planarity. Now, if the molecule is more twisted, especially for the cis structure, it is actually more twisted. If you bring it to kind of, close to an aggregate, the aggregate actually makes it slightly more planar. I mean, we cannot prove it directly, but it seems like that there is a slight planarization which helps the electrocyclization in the aggregated state, and that's why we see a much higher conversion of the cyclized product in the aggregated state. So this is something we observed, and we also figured out another interesting thing is that when you take these photo products, so after photoreaction, if you do for long enough time, this is probably done for 3 hours, you see that the compound, I mean the mixture contains mainly this compound. This is the phenanthine or cyclized product. This is the cis product. This is very less fluorescent. This is known for, so we also have done HPLC separation of all this mixture, and we have separated them and measured their emission spectra separately. And we figured out that this is very emissive, 434 nanometer. This is very less fluorescent, somewhere around 450, but it is very mild. So the emission is mainly contributed by this compound. But if you dry them, what happens? It actually has a very strong quenching in the solid state. If you add the water again, you can get back the thing. So the photo products are luminescent because of this molecule, which is more like a planar chromophore. So the planar chromophore, when you are drying, looks like the dry state it is undergoing concentration phase. Now think about, we started from a molecule which is a twisted molecule. And at the very start of my lecture, I told that we are using twisted molecule because even they go to the aggregated state and solid state, we still have emission. But by photoreaction, I have created more like a conventional chromophore which undergoes concentration quenching in the solid state, but it shows fluorescence in the water medium. We have used that for making a kind of application. So one application we did is called rewritable fluorescent ink. So you see that the light, this word is written here, and under UV light you cannot see anything because in the dry state, this is only from the photo products. But then you add water, you see a strong emission from there. If you dry, again this goes back and you can do this cycle many times. So this is like erasable, and rewritable fluorescent ink. The other thing that we did there's some kind of encryption. So we wrote data, this word, with two different solutions. D and T, these two letters were written in this photo-irradiated product, and these two A's are written with the unreacted cyanosteel. So remember, the unreacted cyanosteel, whether it is the aggregated state or in solution, sorry, the solid state or in the aggregated state, it shows green, yellow emission. So what we are doing here, so if you take this data, which is actually here, if you see under UV light, in the dry state, these two are absent because they have a concentration of 20, and these two are showing the emission color. But if you add water, you can now see all the 4 letters. If you dry again, these two actually vanish and you can get back the color again. So if you want to read all the letters correctly, you need to know what chemical information you are putting to the system. There is a kind of encryption and a decryption required. The same thing we have shown again, you cannot see this is actually white light emitting system and that mixture, but anyway, just assume that we can do also like conversion between white light and the yellow emissive light. So I am almost at the end of the talk. I will finish with something which is kind of unpublished result, but this is something which we observe in the very recent time. So if you think about this cyanosteel bin, I have now talked about them for the last 10 minutes or so, that they undergo this conversion to the cis compound as well as this photocyclization of phenol. But we have never observed anything like 2+2 cycloaddition in this case, which is a bit surprising because if you think about the cell passenger structure, they're still coming close to it. People report that 2+2 cycloaddition is a kind of topochemical reaction where if 2+2 these two bonds have close proximity, you can have a reaction. Looks like when this cyanosteel is packed, they are actually not on top of each other, the carbons are on double bond, and that probably actually inhibits this reaction. We are trying to see that can we actually change the molecular design and induce this 2+2 cycloaddition. What we did is that we made a kind of dimeric cyanosteel bin where 2 cyanosteel bin units are connected through a spacer. So the nomenclature is NMN. So the N, whatever alkyl group is called N, then the spatial length is called M, and then again this side the same thing. The idea is that this molecule, depending on the solvent, for example, in a solvent like DMSO or methanol, where they will be solvated nicely, you stay in a kind of open form. But if you bring water, because we know the cyanosteel bins like to interact with each other every day, they may come close to each other. So as you see from the animation, they may have a folded form. So the question is that in the folded or closed form, can we have 2+ cycloaddition? So of course we did not exactly think it would happen, but just we wanted to see what kind of change in the photoreaction it will bring. So we investigated this compound where you see that this is 8 linkage. So we have taken a kind of 8 carbon linkage which is connecting both cyanosteel bin. And we did the first measurement in absorption spectrum, which did not show too much change, but there was a change in the emission spectrum. If you look carefully, these two emissions are taken in DMSO and methanol, so they are coming somewhere 460 nanometers. And from our previous studies, we know 460, 458 is the range where cyanosteel bins are molecularly dissolved. They are not interacting with each other. But as soon as you take this molecule in water, you see a band somewhere around 510 nanometers. And what we figured out from further studies that this is coming from an intramolecular excimer, especially if you measure that at low concentration. Of course, it depends on concentration because you understand the molecule will keep on increasing concentration at some point. It will also have aggregate formation in other molecules. So in a 0 to 3 micromolar or that range, the molecule is more or less molecularly dissolved. So we are just having one dimer which is surrounded by water molecule. But we see that the 2 cyanosteel bin units are interacting with each other strongly. So this is an intramolecular excimer because if you vary the concentration, this is varying linearly. If it is excimer coming from different molecules, like 2 molecules together, it will not vary linearly. It is only possible. So this range, 0 to 2, 2.5, you have a more like intermolecular excimer. Then we tried to do the photoreaction here because we thought that they may have a slightly different orientation, because now they are connected by a covalent linkage, covalent kind of link. And first we did the reaction in methanol. We figured out that it is undergoing mainly trans-cis isomerization. I will not go into that. We have done detailed NMR analysis. So it probably is a mixture of both, where both the arms are now converted to the Cs, or maybe one arm still Cs and one arm is trans. But the interesting thing which came when we did the reaction in water, because in water, we have seen from the previous studies that they are close, in space in the closed form, and it gave rise to a 2+2 cycloaddition product, because you see a proton coming from a cyclobutane. So this proton, which is also a kind of benzylic proton, comes here, and you see there is only one proton. Why am I mentioning about one proton? Because when you have a cycloaddition reaction, so typically if you bring two carbon-carbon double bond, they can have a head-to-tail arrangement. They can also have something called syn and anti, where the groups are in the same side of that. We have not actually identified. I have just mentioned one product. We are trying to do two-dimensional NMR and also crystallization. But seems like when you do the reaction in this folded state, it exclusively generates only one kind of cyclobutane. And this we have never seen with the single cyanosyl compounds in aggregated state or in solution. And the other thing that was very notable was that the reaction time. So this is just for comparison. You see the previous cyanosyl, it undergoes isomerization and photocyclization. You see after 1 hour you have a 72% conversion. You can follow it from UV and see how quickly it is disappearing. And when you do this 2+2 photocycloaddition, in 20 seconds you get almost 90%. This is extremely now accelerated when you actually make them connected to the linkage. If you see the linkage, it is now having 8 linker, and we also did this with okay. We also figured out that you can also do this reaction with a much weaker light. The initial reactions were done in the photoreactor that we have in the lab, which has a very high power 450 watt lamp. But you can also use a UV chamber, which is more than 1/10 lower power, but still the reaction was quite fast. So this is very good because you can use now lower UV intensity to the same reaction. And what we are doing recently, we are trying to see, because if you see carefully this design, so these two cyanosyls are connected by a reasonably long linker. So if you think about that, there has to be many rotations before they come and form this intermolecular excitement. So we actually initially expected that with a chain link, you will be able to actually control the dimerization cycloaddition reaction, because at some point entropy should take kind of effect, and it will probably not come close together. It will undergo isomerization or other kinds of. But what we figured out, when you have either a smaller linkage or even a longer linkage C12, still we are seeing actually 2+2 cycloaddition product. So this is something which is a bit unexpected, because we thought that at some point it will saturate. We are now trying to make maybe the C16 linker. We want to see that where will be the limit. Of course, we have to understand better that what is that that is bringing the cyanosyl so close to each other so that they can undergo 2+2 cycloaddition. So that is kind of ongoing. This was done by my student Nitish Kumar, and Saurabh has been working on this organic phosphorescence, which I today did not present. And I mainly presented work of Shukla, who just recently did thesis defense. So all this cyanosyl initial work and the initial photoreactions done by him. Rakesh has been working mainly on these pyrene and anthracene based amplifiers and the multi-stimuli responsive phase that I talked about, that was done solely by him. So I thank all of them, because I think, as you see, that the work also needed synthesis and a lot of physical chemistry, a lot of characterization, and it's probably not very easy for students who are just starting in their PhD. So I thank all of them, and also I thank the financial support, ACRB, IISER Kolkata. We recently also got one certificate. And finally, thank you all for your kind attention.
Thank you, Supratim, for a really illuminating talk. And now let's take questions. Maybe I'll first ask those who are online if there is any question, or those who are online.
Yes, I have one question. Hello.
Yeah, please go ahead.
Yeah, very nice talk, of course. There are lots of things actually, and especially I am not an organic chemist, so sometimes I am feeling some difficulties. So first query is that, what is the driving force of aggregation? Is there any hydrogen bonding, or what is happening actually? because you are changing that terminal group, that small alkyl group or large alkyl group, and you are saying that when it is larger, then it has become hydrophobic. So what is actually happening? Why it is aggregating to structure like fiber, nanotube, like that? What is the driving force?
Yeah, so of course, as you see from the last part of the talk, that the cyanophilins like to interact with each other. So they have a kind of interaction, not like planar molecule, but they still like to have a kind of pi-pi stack. But as you mentioned correctly, that for the longer chain derivatives, what we figure that the hydrophobic effect of the chains actually play a major role. Because once you go beyond a particular chain, in this case, from C8 onwards, we started seeing the aggregation. So without, if you have chains below 8, though there is a cyanophilin and other parts are same, but they do not undergo aggregation. So it's a mainly, the self-assembly of these molecules is promoted by the hydrophobic effect, but once they undergo aggregation, then the cyanophilin also can come close to it. So which one is most important, I don't know, but because we see this particularly with a particular, in a specific chain length and beyond, I think the initial stage is set by these hydrophobic groups.
No, but when you are increasing the water concentration, so aggregation is taking place. Is that right?
Yeah, aggregation was taking place at higher water concentration.
Yes,
Yeah.
but if you have a hydrophobic group, then some water, due to some water repelling, some kind of precipitation should appear. Is that a kind of precipitation means that it is forming fiber-like structure?
No,
Or it
precipitate means
is,
you are right. Precipitate can come, but when we make any self-assembly, there is always this chance that if you have very extensive self-assembly, they will come out as a precipitate. So if you control this angle concentration range, so what I have not shown here very detailed, depending on the chain length for C12, the aggregation starts from 10 micromolar. Now, if you take these molecules maybe 200 or 300 micromolar, then this self-assembled network actually comes out of the medium. But in this range, they still stay in the solution and more or less transfer. I mean, this big stars are begin, yeah. So the precipitation, like a macroscopically, if you can see that there is some precipitation only comes at more than 100 micromolar or something. In between, it is still like a soluble aggregate. They are aggregated, but still they are actually held in the solution.
so you showed one image, fiber-like image.
Yeah.
So that remains in the solution.
No, that is actually obtained after drying. So that is why.
No. Before drying, when you are measuring luminescence and other things, it remains in the solution.
Yeah, that time it remains in the solution. So what I believe that this nanofiber network, maybe the dimension that we measure from SCH, that was probably different when we have in the solution. So we did DLS, but GLS, the typical dynamic light scattering, it typically takes the object as a spherical object. So what we did a more like a qualitative comparison, like if you do not have aggregate, you don't see any DLS peak. Then when you have these nanofibers, you see DLS. But DLS cannot measure accurately the size if it is an anisotropic object. So, but that's why I did not tell this is exactly the size, but you can see that some nanostructures are formed.
Yes, that is okay. And another interesting point is that the transformation from the solid fiber to the nanotube, you have shown in slide number 26. So I am curious to know what is the diameter of nanofiber, and then when it is converted to the nanotube. Is there
nanofiber is a very nice question, because this is something we are actually trying to understand in more detail. So nanofibers are slightly higher in diameter, I think close to 70-80 nanometer. But then when they get converted to the nanotube, the sizes were within 40 nanometer. So this seems like
yes, I have seen the scale actually. So one is micron scale, another one is nanometer scale. So I am little bit interested, because if it is a solid material and something from the core side dissolves out, then it will form actually nanotube. So what is happening actually? It is
because the nanotube we are making in a different way. So when you are taking this nanofiber image, this is from the self-assembled structure, and then we dry the thing and then we record the SCM. When you are making nanotube, we are adding actually heparin in the solution phase. So it seems like what I have not shown that we have taken some, in between some images like where you add small amount of heparin. It seems as soon as you start heparin, these nanofibers start breaking down, and it forms some kind of, some cases, sheet-like structure, which probably fold at later stage. So the things are happening in solution, but what I have shown in the final stages after dry, but heparin is not added to the dry nanofiber, is added in the solutions.
Yeah, there are lots of things could be happening.
So I think this is something, probably if we had the chance to do cryo-EM, maybe we could trap some of these structures, but unfortunately we are not able to do that level at this point. Maybe in future we'll try to do. Because this is something interesting, that how heparin is actually converting from a solid nanofiber to this hollow nanotube.
Yes, very interesting that observation. And another, just last my last question is that the white light formation. You have shown that ZSCN is transforming to ESCN. That is a permanent change or reversible? First of all, I'd like to know.
Yeah, so that part is actually reversible. So, but what happens? Yeah, that is reversible. But
No,
if you,
that is also happening by photocycloaddition, isn't it? Because in the
no.
ZSCN it is a planar, and in your ESCN some anthracene type 3 benzene links are together.
Yeah, I understood. So let me just elaborate a little bit. So
this is reversible.
I know, I'll tell you. So generally, if you irradiate J, it first goes to E, so trans to C. So this part is reversible. But then, when the next step, electrocyclization happens, then there is a irreversible oxidation because we are doing under ambient condition. So the final stage, the financing formation is irreversible. So the other steps are actually reversible. But the final stage drives the equilibrium to the photocyclization.
And that white emission is originated from which structure?
White emission was actually this actually originated from a mixture of two emission colors.
Okay.
The unreacted trans, it gives greenish yellow emission, and then when you make this financing, which shows actually cyan blue. So at a particular time when the emission colors are perfect, they will give the white. And if you continue, it will go more towards the blue, cyan blue.
Yeah. So white color can be retained, that white emission.
That white emission, what we have seen, if you keep it dark, I think it was stable more than 7 days. We did not check. So we also checked the UV and fluorescence. There was only minor modulation. Okay. Under ambient light, there is some change. So maybe 2 to 3 days you can keep. It's not having very bad stability. I mean, not very high stability, but it is reasonably good for a few days.
Okay. Now let's go.
Thank you.
Yeah.
Thank you.
Nice.
Thank you, Professor Dey. Any other question? Yeah, Pradeep.
I have one question. Most of the molecules what we was showing, they are very similar to halogens. Use the group of resistor.
Yeah, I know halogens. Yeah, but the halogens, what happens? The halogens, when you connect with two pyridiniums directly, so halogens typically it does not have luminescence. So, but nowadays, as you mentioned, I will tell you, several groups have reported extended halogens. So they start getting luminescence. So there is some difference with that. So in case of those compounds, so once you introduce this cyano group, it has some typical nature. Of course, the extended halogens also will have a twist, because once you have a biphenyl connection, there will be always an inherent twist. The twist becomes more in this case because we introduce cyano. So it twists one part, there is a kind of result, the other parts also start getting more twist.
In this study, halogens has been incorporated cucurbitacin almost.
Yeah, that is one of the classic studies. So cucurbitacin length actually matches very well with the biology.
So you have done on there with your system.
My system, yeah, I have not shown, I think, in detail that host gas chemistry. So our molecules also go inside. Actually, I have this extra slide. Do you want to see or no?
We will see that.
Yeah. So I have not shown in the main slide. That is in the extra slide because today I did not talk about host gas chemistry.
But it's done on.
Yeah, it's done. You can do NMR, you can do isothermal titration. Calomel, we have a very high binding affinity. So we have done that time with the CB7, so which is having actually 7, like a 7-member oligomer of this family. So it goes very nicely and it shows nice change in the fluorescence.
Yeah, that would be the way to avoid this cycles at one point of time. Like other ways,
very good. So we have already done that. I have not shown. So if you add cucurbitacin 7 and if you have this, I thought that this dimer, because they are, so if you block one of them with CB7, the 2+ to stop. So we have already done this control study to show that this intermolecular exciter formation and this this proximity is very important. So you can destroy the proximity by kind of trapping one of the arms with a glue set. Yeah.
Any other question? Yeah.
I want to ask that what is the solubility limit of the cyanosilvine?
So that depends on this molecule, because we have introduced this to pyridinium at the end, and the limit is also determined by the alkyl chain. So if you take the C2 compound, the ethyl analog can go very close to, I think, more than 1 millimolar. But if you go for C10 or C2, generally more than 100 micromolar, they will start precipitating, because they are like they do extensive aggregation, so they will come out as some.
In that case, the luminescence happens in the solution phase, but not in the solid. Okay, so
No, that depends. That's probably so that was for the photoreacted mixture. For the cyanosil means you have a solution phase, aggregate state, high luminescent, and also in the solution. For the photoreacted mixture, because you are generating financing, it seems like that is undergoing typical concentration.
comes due to the extensive conjugation.
But extensive conjugation is, if you look at these twisted molecules on paper, they have extended conjugation. But I will tell you a very simple thing that we think. So if you look at the absorption spectra, I have shown somewhere for this cyanosil, you have almost 11, 12 double bonds in conjugation. 11, 12 double bonds in conjugation is also there, for example, beta carotene, which is a more or less planar structure, that is there in tomato. So carotene actually absorbs close to 500 nanometer, because there the backbone is planar. So in this cyanosil, though they have equal number of double bonds, because of the twist, you have only local conjugation. So 360 nanometer, I think that will translate to 5-6 double bonds in conjugation. So this looks a very long molecule, but actually it is like locally conjugated, not throughout the back.
There is no technique.
No, we have for the cyanosil means in the solid state also you have strong emission.
In the reaction mixture.
Reaction mixture is different. Now reaction mixture, you are actually converting the cyanosil. Cyanosil is no more. No, cyanosil means you have a double bond, both sides phenyl. Now we actually go to a phenanthine, which is a different molecule. It's a different aromatic molecule.
Yeah. Okay, so for the interest of time, let's close here. And if you have further questions, you can always discuss outside. So let's thank Supratim once again for a nice talk, and please join for tea outside.